Good evening, everyone. Welcome to Botany Insiders, another free class. Let's wait for the students to join in and then we'll start off with our class. Okay. Till then, please let me know in the chat section whether the audio and the video is clear to you or not. Are you able to check the presentation? Are you able to hear me properly? Just let me know in the chat sections, everyone. Let everyone join, then we'll start with our class. Okay. Good evening to all the students. Okay, Mayank. Others also participate in the class. Just confirm whether the audio and the video is clear. Perfect, perfect. Thank you. So we'll be discussing about the GPCR pathway in detail. Good evening, Shiva Priya. Okay, perfect. So we'll be discussing about the GPCR pathway in detail in this particular class. Okay. So before starting the video, if you're new to Botany Insider, please make sure to hit the subscribe button. Also, we have 3 full-fledged contents available for you if you are targeting the December examination. We have our own application on which we conduct our classes. Okay, we have a full course, we have a crash course, and we have a notes booklet available for you, hard copy, which will be delivered to you at your doorstep if you place your orders for the same. Okay, so let's begin. In addition to that, we are about to launch PYQ course for the December 2023 students, where we are going to discuss all the questions from 2023 up till 2016 or maybe below before the years, depending upon the timings. So we'll be discussing all the questions in detail, in depth. Okay, so let's begin with our class now. Right, if you remember yesterday we discussed about that what is cell signaling all about. I'm expecting that what cell signaling is clear to you. I have made an entire playlist, like every day, whatever class we are going to discuss, you'll get that class in that playlist itself. It's with the name of December 2023 free classes. Okay. So try to like follow that class if you are like sincerely preparing and if you want to get through each and every topic. Okay. So in the end of yesterday's class, I gave you an entire list of the topics, right? So now we are going to discuss about GPCR. If you have seen the previous year questions, you can understand how important this topic is. Almost every year, you can find a question related to the G protein coupled receptor. And in this class, I'll give you a detailed understanding of what this complete topic is all about. Okay, so let's begin. GPCR, it stands for your G protein coupled receptors. First thing first, GPCR, it stands for G protein coupled receptor. So, what is this complete signaling all about? So, it's a large family of your cell surface receptor which have a very, very important role in what? It have an important role to transmit the signal from external environment of the cell to the internal environment of the cell. Okay, this particular receptor helps in transmitting the signals from the external environment to the internal environment. Okay, now we'll be understanding about the entire topic in depth. Okay, so understand this particular thing properly. Okay, so when we look into the G protein, focus upon the presentation. Okay, first we'll understand about it in depth in the written part and then I'll explain you with the help of theory also. When you look into the G protein, so it's a trimeric protein. Okay, there are 3 segments to it. There are 3 subunits to it. What are the names? Alpha, beta, and gamma. The G protein is having 3 subunits: alpha subunit, beta subunit, and gamma subunit. Okay, it is arranged in this particular orientation. So alpha, then you have beta, and then you have gamma subunit. Okay. Initially, what do we have? Initially, when the G protein coupled receptor, when the receptor that we have, which like I'll be explaining that thing as well. Wait. When you look into the receptor, so it's a serpentine receptor, which is having the external site and which is having a cytosolic site also. This is your plasma membrane. Okay. So on your plasma membrane, it's a serpentine receptor. That is, it will span your plasma membrane 7 times. It will cross your plasma membrane 7 times. Till the time there is no ligand over here, at the time when the ligand is not available, your G protein remains in inactive format. Okay. So our G protein is inactive when there is no ligand, okay? When the ligand is absent, right? As and when the ligand binds to it, your G protein gets converted into the active format. How does it get converted? Understand this particular thing now. So during the time when there is no ligand, at that time, all the three subunits, that is your alpha, beta, and your gamma, will stay in this particular orientation. As and when ligand comes and binds over here, so what will be observed? This alpha subunit will be getting dissociated, and your beta and gamma unit will stay here itself. And the reason for the same is because initially what happens, you have your G protein initially bound to your GDP. Okay, it is initially bound to your GDP in your inactive format. When it gets converted into, or when the ligand binds to it, when the ligand comes and binds over here, it gets converted into the active format. How? Via conversion or via the GDP getting exchanged with GTP. Over here, you have the utilization of one important factor, which is given the name of guanine exchange factor. Very, very important. Okay. So when the ligand binds over here onto the cytosolic side, onto your on your extracellular side, onto the cytosolic side, your guanine exchange factor will help in the conversion of inactive G protein into your active G protein. Okay, with the help of guanine exchange factor, which will help in the conversion of GDP into your GTP. Okay, now your G protein is in the active format where you have the alpha, beta, gamma subunits onto the say, like initially they are all together. When the guanine exchange factor is helping in the conversion of GDP into GTP, your alpha subunit will be getting dissociated from this orientation, and your beta and gamma will stay in the same place. Okay. So your alpha subunit will then be helping in the various other processes. What is the entire process? I'll make you explain that. Before that, you need to get this thing clear, that how does your G alpha subunit is getting activated. So what are the events happening? You have the ligand binding, you have the cytosolic site transmitting the signal to your protein, to your G protein, which is initially inactive because there is no ligand. It will be there in your alpha, beta, gamma subunit association. As and when the ligand binds, it will transmit the signal. GDP will get converted into GTP with the help of guanine exchange factor. Your G alpha subunit becomes activated, and it will be further working for the next steps, right? So till here, just let me know whether the things are clear to you or not, because after this, I'll explain you that after the activation of your G protein, what is the next step? Okay. Quickly get through this. If it's clear, then we'll move forward. Okay. Quickly let me know about this, whether it's clear or not. Yes. Clear till here, everyone? It's a very important topic, okay? It's a very, very important topic. So pay attention to the entire class, right? 23 of you are there in the class. Please make sure that you are participating, right? Now, after this, what happens? After this, your G protein is now active. Now, the main step, very important step. Understand this, okay? And this complete thing is occurring in your plasma membrane itself, okay? This complete thing is occurring in your plasma membrane. I'll show you one diagram also. So just understand it here. This is your plasma membrane itself. Just to avoid this confusion, I'm writing it here. Now what happens? Your G alpha subunit, which is bound to your GTP, it will help in the activation of your adenylate cyclase, AC. It will help in the activation of your adenylate cyclase. Okay. As and when the adenylate cyclase gets activated, adenylate cyclase will help in the conversion, or it will help in the activation or conversion of your ATP into CAMP. CAMP's basic functionality is that it is your secondary messenger. Okay. ATP gets converted into CAMP. Now, most important step, your activated CAMP will be converting, or it will be activating your pKa, that is your protein kinase A, from the inactive state to the active state. Okay. From inactive state to the active state. How does it occur? Let me draw it on a next slide. It will be easier for you to understand, right? Now, what did we understand till here? You have a serpentine receptor, N side, C side. This is your entire plasma membrane, right? This is our plasma membrane. Here we had our G protein in the alpha, beta, and gamma subunit, right? Now, with the addition of your ligand, your GDP got converted into GTP with the help of guanine exchange factor, which helped in activation of your G alpha. Now, this G alpha helped in the activation of your adenylate cyclase, which helped in conversion of your ATP into CAMP. What is it? It is your main factor. It is your main secondary messenger. CAMP will be converting pKa, protein kinase A, from the inactive state to your active state, right? Now, from inactive state to your active state, this occurs with the help of four of the subunits of CAMP. Four subunits, like four of the molecules of your CAMP is required for the conversion of inactive format of your protein kinase A into the active format. If you look into the structure of your protein kinase A, we'll be discussing about this in depth also. Just understand for the point time being. In the protein kinase A, if I show you the entire structure, so look over here. This is how the protein kinase A actually looks like. Okay. In the protein kinase A structure, you have two of the catalytic subunit and two of the regulatory subunit. Catalytic subunit does not have the cyclic AMP binding site. It is the regulatory subunit which is having your main site where your cyclic AMP can bind. Okay, so two regulatory subunits are there and two catalytic subunits are there. On to your regulatory subunit. Per subunit is having two of the CAMP binding site. One regulatory subunit will bind to two and the second one will bind to two. So in total, four CAMPs are required for the conversion. Okay, for the conversion, four CAMP will be required, right? That's the reason why we have four CAMP required for the same. Okay, now, so over here you had four CAMP required for the entire process for the conversion of your pKa from the inactive to the active state. Okay, now what happens from your inactive state to your active state of pKa? Initially over here it was the plasma membrane, right? Now this pKa will be entering into the nucleus. Okay, now this pKa will be entering into the nucleus. Consider this is your nucleus. Okay, in the nucleus, this pKa, after entering, it will be activating your CREB. What is CREB? CREB stands for Camp Response Element Binding Protein. Okay, what is CREB? It is CAMP, Response Element Binding Protein. Okay, so this pKa will be helping in this activated pKa will be helping in the conversion of your inactive CREB into the active CREB. Okay, from your inactive state to your active state. Now this CREB will be joining with your CBP, that is CREB binding proteins. Now, there's a complex formed between CBP and your CREB. These two join together. They both will be targeting a specific transcription factor and the response will be seen in the cell. Okay, this is how the entire functionality occurs. Okay. So last step is CREB converting from the inactive state to the active state with the help of your pKa, protein kinase A. CREB joins with CBP, that is your CMP binding, CREB binding protein. They'll be activating a specific transcription factor depending upon what the ligand was. Over here, the ligand was there. Over here, you had your G protein. It got activated alpha, right? It activated your ATP into CAMP. 4-CAMP helped in the conversion of the same, right? We just had a discussion related to the same. So depending on what ligand this particular complete pathway will be receiving, in the similar orientation, you will be having the response T. But this is the basic pathway, how the complete GPCR actually operates. Okay, quickly understand this, everyone. If you find any difficulty, you can ask. Otherwise, I'll show you the entire like my own notes which I made during my preparation time. I'll show you those notes. Understand this properly. It's very interesting and a very important topic. Okay, go through this, everyone, and if you find any difficulty, ask in the chat section. Are you understanding the entire concept behind the same, right? I'll show you the structure of this protein, this GPCR also, and the entire functionality also. Clear, everyone, till here? Just let me know in the chat sections, everyone. Here, very good, very good. Okay, now let me show you the entire, like my self notes which I made during my preparation time. See, understand this. See, whatever I have explained you, exact same thing is written. So you have your GPCR protein over here. The GDP gets converted into the GTP with the help of guanine exchange factor. Alpha, beta, gamma subunit is there. G alpha, subunit will be getting activated when the GTP is bound with the help of guanine exchange factor. Guanine exchange, this G alpha will be helping in the conversion of your ATP into CAMP via the activation of adenylate cyclase, right? Adenylate cyclase will convert ATP into CAMP. 4 units of CAMP required to convert pKa inactive to active. pKa enters into the nuclear pore. In the nuclear pore, pKa will be activating CREB. CREB binds with CBP to activate targeted genes, which could be CAMP response elements. And finally, the response is seen depending upon the ligand which gets bind to it, right? So CREB is what? CAMP response element binding protein, which I just told you. CBP is CRB binding protein, which I just told you, right? This is like directly from my notes, which I use during my preparation time. Okay. Quickly understand this, read this, and if you find any difficulty, you can ask. Understand this entire thing, everyone. Very, very important. Okay. This is just the same thing, exact same thing which I just told you over here. Okay. Exact same thing is written down here. Right now, let me explain you the structure of the G protein as well. Okay. Understand the structure of the G protein. See, what is GPCR? It is G protein coupled receptor pathway. It's a serpentine. It spans the entire plasma membrane 7 times. It is heptameric in nature. Then you have alpha helical structure. So when we discuss about when you study your biochemistry protein structure, in that, in the secondary structure, like there are 4 structures in the protein structure: primary, secondary, tertiary, and quaternary, right? In the secondary structure, you have alpha and beta subunits. Sorry, you have alpha helix and beta sheets, right? Kamini, just wait. Let me complete this first. Okay. So you have the N terminal, you have the C terminal. So over here, this G protein, entire G protein is what? This G protein coupled receptor is made up of your alpha helical structure, right? So it is having 7 of the domains which is crossing. So for prime 1, 2, 3, 4, 5, 6, 7. 7 of them are there. This is your external region. This is your internal region. Then you have the plasma membrane, okay? The main plasma membrane region, okay? So you have first loop, second loop, third loop. You have first transmembrane loop, second transmembrane, and the third transmembrane. In the third transmembrane loop, you have the ligand binding site, right? In the third loop, this will be joining to your G protein, okay? This G protein is this, and after the binding of the receptor ligand and then the entire process, so this is how the same thing will be occurring. This is your structure of your G protein, okay? The entire structure of your G protein. Go through this, everyone. Very important. I'm telling you, if you go through this 30 minutes properly, you can solve all the questions which are there from your uh, main thing which your from your GPCR basic questions. Okay, we'll be discussing few questions also. Like just after this, I'll show you one question which you can practice out also. After this topic, let's discuss one question also. Kamini, PKA is protein kinase A. So in that you have the inactive form to the active form. It gets inactive to the active with the help of 4 of the CAMP proteins. Okay. So 4 of the CAMP molecules. So it helps in the entire processing. Okay. Clear everyone till here? Right now I'll be explaining you the entire, like the one complete question we'll be discussing. First, you will be marking the answer and then we'll be discussing the solution for the same. Okay. It's the latest year question. So let's see how many of you can solve it out. Till here, I'm expecting the concept is clear to all. Right, let's discuss the question now. Just a second. Let me show you the question. Okay, this is the question. Give it a try to the question, everyone. What is the question all about? Question is the cycling of the monomeric G proteins such as RAS between the active and the inactive states. One more point. Remember one more segment. I forgot. Like over here, remember this active format gets converted back to the inactive format with the help of your GAP. Okay, very important. Just remember this additional point from your active format to the inactive format, because there need to be some regulation also for the G protein activation. So for that, GAP. So G alpha inactive to active is via GEF, and active to inactive is via GAP. Remember this, just one additional pointer. Okay, I forgot in the previous one. Remember this. With the help of this, try to find the answer for this. The cycling of the monomeric G protein, such as your RAS, between the active and the inactive is with the help of your GAP protein and your nucleotide exchange factors, guanine nucleotide exchange factor, GEFs. Following conditions are given to you. Following conditions are given to you. You need to find out the correct answer for this particular question. Which of the following option represents the state that might cause a constantly active signaling cascade? Okay, this is a latest year question, and if I'm not wrong, this one is from June 2021. Okay, this one is from June 2021 or 2022. I'm not like complete sure about the year, but it is from either 21 or 22. Give it a try to this question, everyone. And if you have understood the entire concept which I've explained you now, you can easily mark the answer for this. It's a 4-marker. Kamini, try to adjust in this, okay? Try to take a screenshot and then read the question properly, because this is the maximum size of the question which I can put in. Get the answer for this question, everyone. Try out, everyone. Very easy. Just remember that G active, like from the inactive to the active state, it is via GEF. From your active to the inactive state, it is via GAP. Remember this concept and you'll get the answer. Everyone participate. 25 of you are there in the class, and I want each and every student to participate. Very easy question, very easy. It is asking us which of the following option represents the condition that might cause a constantly activated cascade. We need to keep the protein activated for the entire pathway, for the entire concept. We need to keep it activated. Okay. Everyone participate in the class. 26 of you are there. 7 to 8 have marked the answer already. Okay. Mark the answer for this question. Understand the concept. Very good, very good, very good. Excellent. That means you people are concentrating in the class. The first option is a non-functional gap. That is, if I have a non-functional GTPase activating protein, can I keep my cell, can I keep my protein in the constantly activated state? Yes, for sure. So, first option is correct. If my first option gets correct, I can eliminate B and C. Now, read the second one. Permanently activated gap. This one is incorrect. Reason being, if gap gets activated, then it will be converting into the inactive state, right? Then you have non-functional GEF, that is guanine exchange factor becomes non-functional. How will it get converted into the active state? This is also incorrect, right? So I can eliminate A, like B also is incorrect, so I can eliminate the first option also. And with this, I have reached to my correct answer. A and D. Excellent. All of you, those of you participated in the class, all of you got the concept clear. So a permanently activated GEF, that is GEF is permanently active and your gap is permanently inactive. I'll get my cell or I'll have my constantly activated signaling cascade. Okay, answer is option number D. Fourth is the correct answer for this particular question. Is this clear to everyone? Yes, in this half an hour, I've tried to give you the understanding of one of the most important concept from cell signaling cycle concept. The complete unit is important, and if you look into various pathways, your GPCR is one of the most important pathway, and I am expecting that it's clear to each one of you, right? Just let me know in the chat section. Was this class helpful? So tomorrow, let's take up one more signaling pathway, right? Okay, so let's meet in the next class now. I hope things are clear. And if you feel like that this particular session or these classes help you out, we have a full course and a crash course, like a complete structured 3-hour sessions goes on every single day in each of the course. So you can download our application and get yourself enrolled in the same. Okay. For the time being, crash course is running, full course is running. You can also order your notes booklet directly via WhatsApp also. You can WhatsApp us on this number. You can WhatsApp us on this number for any of the queries. Okay. All the best, everyone. Bye. Take care. Let's meet in the next class.